Okay, and one more person is joining us. Okay, so welcome to D-Challenge 2022 Kickoff. Oops, excuse me for that. And as you can see here, with the support from the, we're presenting this with Sugar Science and dkNET and the GADRF T1D Fund. We're also gaining support from T1D Knowledge Portal, HERN, the Human Islet Research Network, and leading T1D Sciences as judges. I'm going to go through the presentation, and then at the end, if people have questions, we're happy to answer them. Okay, so what is the D-Challenge? Well, it is a collaborative effort of sponsors and promotional partners, as well as resource portals and workspaces, some of which are shown here. You can see there's a wide variety of these resources, and most all of them are accessible via the dkNET portal. Funding for D-Challenge is provided by the Sugar Science and dkNET. And what is the Sugar Science? We help the global T1D research community connect. Our efforts thus far have brought us a scientific audience of 35,000, and we continue to grow. So what is the D-Challenge? What is it actually all about? So it's really an in silico-based competition. Scientists and trainees participate by mining rich data sets. Many of those are accessible through the dkNET portal. And our ultimate aim is to create a novel hypothesis or hypotheses pertaining to type 1 diabetes. We held the first one in 2021. It was successful, and now we're holding the second one. And so what are we asking participants to do? We want people to use data-driven resources and tools to develop a compelling hypothesis about the biology, treatment of sequelae of T1D. We're asking them to use partnered resources to mine rich data sets and develop viable hypothesis for type 1 diabetes. These number three, the novel hypotheses will be pitched to JDRF T1D Fund in a pitch polish setting, and finally to our community of judges, and prizes, cash awards will total $50,000. And we really encourage open science principles. For example, the formal pre-registration of any hypotheses developed will work with the winners to publicize their contributions at a high level. This year we added a novice track, so trainees are invited to participate in the D-Challenge and develop a novel hypothesis from rich data set. As novices, participation in this track requires attendance in a series of D-Challenge bootcamps, which I'll talk about later, and they will be rewarded by a $100 Amazon gift card. We've had feedback from some of the trainees and people that want to participate in this novice track that they would also like the opportunity to develop a hypothesis, and so that will also be considered. So we're asking people to really think way outside the box, right? We're not asking you to have a full proof of concept, but really to develop a hypothesis from which you can launch your further inquiry. And see, these are some of the logistics. We have judging and prizes. How this works is that on November 30th, each team submits a novel T1D hypothesis and full documentation. November 9th, finalists are announced. December 12th, those finalists will present a pitch polish to JDRF, the T1D Fund. For those who don't know, they are a group of venture capital funds, fund managers who decide where to invest monies in type 1 diabetes focused business ideas. So it's an excellent opportunity to gain connection to these individuals and get feedback on your actual idea. December 13th, we have a live pitch to judges, and then the 15th, we have an award ceremony. You can see how we've broken out the prizes here. First, $25,000, second, $15,000, two standout prizes at $5,000, and then $2,100 novice track cash awards. The judging criteria you can see is, I'm going to try to move this over. I'm not sure if you guys can see this. Impact of hypothesis on T1D research, effective use of diverse online resources, feasibility of proof of concept, and complete documentation. Okay, here we go. So if you want to really do this thoroughly and properly and correctly, you really need to show your work. That we really do ask that you show how you got from point A to point B in your thinking, how you developed your hypothesis, and which data resources you used. So let's start with how does it work? So we provide a starting point that the registered teams use to design this working hypothesis to further research in the realm of T1D. dkNET has an incredible data-driven environment that allows investigators to dive into data and formulate new hypotheses. You are also welcome to use your own data resources or tap into data resources that will be highlighted to you by a variety of experts in the boot camps. So what should teams do? What should they be thinking about? You should be using any available data sets to think about pathways to create novel hypotheses, as well as the dkNET portal as a gateway for locating other online resources, data sets, tools, services, possible reagents that provide supporting evidence for the hypothesis. You should also use multiple online resources. Really want to steer clear of avoiding heavy reliance on the literature. You don't really want to just sort of like comb through the literature and come up with hypotheses. That's not the focus of this challenge. So we really want to see breadth and depth of engagement with online tools, their use, going way beyond sort of the Google and PubMed, high level inquiry. And we'd like to see it carefully document, the teams to carefully document their approach and data. And so we'll sort of showcase this in these boot camps to show how, you can use dkNET for resources and data management and FAIR. We are asking you to explore the space, right? You can see there's all these different resources at your disposal. And they all, there is some overlap, but they all have unique features. So you can really kind of dive into these, according to your current expertise, or if you want to extend that expertise, you can, learn more about these individual resources and drive it from there. So what are the goals of the D Challenge? Again, just to sort of tap this one more time, we'd like you to develop a compelling hypothesis about the biology, treatment, or sequelae of type 1 diabetes and increase awareness. Share this experience with colleagues that there's a rich ecosystem of online tools and data to be used and had. Again, projects have to demonstrate the value of public data and/or demonstrate the value of public online bioinformatics tools and/or demonstrate fairness of your approach, FAIR. Jeff can talk about that a little bit more. In terms of what the teams should look like, you should have a lead in your team. Some teams are one person, so then you're going to have to wear all three hats. But you should have a lead, which a person who understands the science and assigns the roles, sort of like the director. Then you have a team admin, decides where the data and tools go, deals with, in conjunction with technical assistance, any technical issues. That's sort of the tech lead. And then finally, the writer, who really kind of leads the way in terms of your documentation process. You'll have a ability to use all the relevant software and technology to communicate and share information. You can use GitHub. We'll have a Slack available. You can use Google Drive. You can use email. And we're going to, during week 1, we're going to set up and invite you into Slack and into the Google Drive. Okay, and then we've got a little bit about relevant software and technology. And I think this is where, yes, this is where Jeff dives in.
Yep.
So welcome, Jeff Frith. He's needs probably no introduction, but he's deeply involved in dkNET and located in San Diego at UCSD. He has a long interest and a wealth of knowledge in data and bioinformatic data. Thanks, Jeff. Can you take it away here?
Thanks, Monica. So as Monica had mentioned, one of the key things that we wanted to get out of this challenge is, to sort of, again, highlight, the usefulness of, online, data resources, online bioinformatics tools that are being, increasingly being made available to the community. And to help with that, one of the things that we want to do is to also, provide from the teams, sort of the documentation of, the resources and tools they used to come up with their hypothesis, because there are so many that are out there. This is to help document what you use, but also then to help inform dkNET and others about, what tools are available and their usefulness. So one way that we're doing this is to introduce everyone to another tool, which is protocols.io, which is an online system to basically document protocols. So you can document step-by-step protocols within protocols.io. It is free to sign up. You're given a free number of sort of private protocols that you could generate in protocols.io. So this is one of the ways that we're also trying to help the community get engaged with making protocols more reusable and shareable. And so there are a number of projects that have begun using protocols.io to document protocols. So next slide, Monica.
Oops, sorry. There you go.
Okay. The other thing that we are promoting the use of is also the use of research resource identifiers. So if you've published manuscripts, in the last year or so, you've probably been asked to provide these identifiers for some of the resources that you used. And so basically, what research resource identifiers are persistent identifiers for research resources, so that it's easy to basically ID and also discover uses of these tools, reagents, antibodies, cell lines within the literature, to make it easier for the researcher later on to find out, for example, where has specific antibody been used, for example, if they're trying to find out if there have been any issues with the antibody, or similar studies that have been done using that antibody that may help, in your current study. So that's, why the RIDs are being, suggested by a large number of journals. And we also request that, when you're filling out your protocol, that you use these identifiers to tag the resources, that have been used. Next slide. So a little, give people a little intro to dkNET. So the NIDDK information network, is funded by NIDDK at NIH and is meant to be a portal to provide information to researchers around data sets, tools, and also issues around data sharing. Next slide. So, when NIDDK had funded this, it was funded as a research resource information portal, and a way, to connect, researchers with resources that are out there. Again, there are so many of these resources out there. Sometimes it's hard to know, what is available. I mean, there are a few very well-known resources, but there are also a lot of other resources that are incredibly powerful, that you may not be aware of. So this is funded again by NIDDK, and it is developed and maintained out of the lab I co-direct with Marianne Martone at UC San Diego. Next slide. And within the portal, there are 5 basic areas. One is research reports, which help researchers find and evaluate resources, and this is tied into those research resource identifiers. There's a section on authentication reports, which tries to help aggregate information and help provide information for the NIH authentication plans. We have a set of services that allows you to search across different repositories, to different databases. And then we also have tools and also information where we're trying to help people deal with data sharing mandates, especially with the new 2023 NIH data sharing mandate that's going to require all researchers funded by NIH to share all data that they have collected, even if it has not been used in a published paper. So even data that may be considered negative data or dark data at this time will have to be published for any grants submitted after January 25th, 2023. And then we have a hypothesis center where we've brought together some initial set of tools such as signaling pathways project and others where we provide some tutorials to help researchers gain access to bioinformatics tools that are available online. Next slide. And so again, if one looks at sort of the overall sort of life cycle of research, dkNET is trying to help researchers at multiple stages from, the background research, gaining information, potentially constructing hypotheses through some of the tools in the hypothesis center. When you're planning your experiments, looking at resource reports, to find out information about antibodies, model organisms, cell lines you may be using, all the way through publication of results, again, where you will need to get, the RIDs for, the antibodies, cell lines, organisms you're using, plus also the resources around FAIR data and, the tools, steps needed to make sure your data is properly shared with the community as per NIH mandates. Next slide. So this is just an example of a resource report. So again, these are tools that are available. So you can go in, you can search for example, software tools, databases, and then you can get a full, you get an individual page for each resource that provides information on the resource, but also provides information on any citations of that resource that we have been able to pull using the resource identifiers in the open access subset that we're actually allowed to text mine. So that's just a little overview of the resource reports. Next slide. Okay, the discovery portal, again, is also a search interface, but here we look into some of the data resources that may be available. And so it provides a quick overview of where resources may be available. We don't, we're not able to cover every database that's out there, but as we learn about new databases, we do try to incorporate them. Next slide. And then the hypothesis center. So this is, example of some of the tutorials, that we, have developed. Here is a tutorial that links, information pulled back from the mouse metabolic phenotyping centers and then pipes that through the signaling pathways project to explore, some phenotype genotype associations. Next slide. And so again, we do know that there's a significant growth of available data and online tools and resources, and that's exactly why we're running this specific challenge. Next slide. And there, within the type 1 diabetes realm, there are a number of specific tools and resources specifically targeted to type 1 diabetes, and then there's a broader range of tools and resources that are more generic in the sense that they cover more than one disease. Next slide. And so again, there are a number of these rich resources. The resources that we've been showing, like the type 1 diabetes knowledge portal, Pancreatlas, PanCD. These are going to be in upcoming boot camps, and then also through dkNET, through our YouTube channel, you'll be able to see a number of webinars or tutorials about these resources, that we've already, had webinars for. Next slide.
Sorry, it's a little stuck. There we go. There's the webinars.
So these are the video and webinars. And Monica, I believe that you're also going to be making the boot camps available online, correct?
Yes, those will be released this afternoon.
Okay. And then as the new boot camps are done, over the next 2 months, we'll also be posting those online.
Right.
Next slide. I think we're close to the end. Yep. And
yeah, so these are some of the important dates for you guys to highlight, and we'll be sending out an email after this to reiterate these. But basically, today's the kickoff, the first boot camp. And then the next one comes up on September 23rd, which is Pancreatlas PancDB, presented by Golnaz Vahidi. She's one of the founders and managers of it at UPenn. September 30th, a huge AMP T1D knowledge base that will be presented by Kyle Galton and Noel Burt at UCSD and MIT The Broad, respectively. October 7th is going to be the Hearn Resources and CalIR, presented by John Cadiz and Alex Banks. October 14th, we have NPod and Stitcher, presented by Mark Atkinson and Jamie Heather. And then October 21st is AIRR and Navigating iReceptor and Autoimmunity from Brian Corey, and likely with Felix Baden, one of the founders of the AIR, or the Antibody Society. And then October 28th, we have sort of, again, coming back to the dkNET world, resources and fair data for 2023 will be presented. And let's see if I can move this around a little bit. We're asking that a minimum of 1 member per team must attend check-ins. And then, then some big picture dates are November 18th. We'd like you to submit your abstract, and we'll give details about what that will entail. As we get closer, November 30th, submit your final hypothesis. December 9th, finalists will be announced by the judges. December 12th, we will have the pitch polish with JDRF T1D Fund for the finalists. And then December 13th, a live pitch to judges. And finally, the award ceremony. And we have a number of judges we'll be compiling once we see what the interests are, for the participants. But as of right now, we have Steve Rich, who is a very well-known bioinformatic geneticist from UVA. And we also have Ben Busby joining us from DNA Nexus. He's also former NIH. It looks as if we'll have Noel Burt and Nina Luning-Prack. Noel Burt from MIT The Broad. She's one of the co-founders of the huge AMP and the T1D Knowledge portal. And Nina Luning-Prack is an MD PhD from UPenn, and she heads up the HPAP there. And finally, these are really sort of like, some of the best and the brightest in the particular field, in their particular field. So it should be great to connect with them, as well as, gain, feedback from them during the course of the D challenge. So I'd just like to open it up to questions at this point. If anyone has a question, please feel free to try to open this up a little bit. Here we go. Okay, if you have a question, just raise your hand or unmute yourself. We're happy to answer any of your questions.
Hi, Monica. Can you hear me?
Yes.
Hi. This was really great. A lot of great resources. I have a question. If from last year, is it possible to see sort of the winning proposals and basically kind of, what a good and a winning proposal looks like, if that is available for us to see?
Jeff?
Yeah, I mean, the detailed proposals, no, because they're being turned into research proposals. I mean, that's one of the goals, of the challenge, was to, provide, some money to get some potentially to build out the hypothesis. But we do have, Monica, we do have the public pitch, the public pitches
yes, we have the public pitches available, and we'll give you access to those. Also, one of the participants ending up as a participant, Neil McKenna, he did take some of the findings of working through this D-challenge and turned it into a paper that is public now. It's been published, and it's an excellent resource to sort of see what you can do with sort of going back through data and looking at it in a new way. And we'll provide that to you guys also on Slack. But that's a recent paper, and it's very interesting. He looked at PBMCs through the and reimagined the data and was able to sort of identify some genes that were up-regulated and down-regulated in context of type 1 diabetes.
Yeah, and so I think, even though we can't share some of the specific details, the main criteria, again, that the judging is focusing on is, one, that it is a testable hypothesis, right?
Mm-hmm.
So, that whatever is hypothesized is something that can be taken to the laboratory and tested in some way. That you do use a collection of resources. There was sort of difference in terms of what resources people used last year, where some people used a broader collection of online resources, and some groups used sort of more limited resources, and some that were sort of harder to gain access to. So we are looking to see use of sort of a collection of these online resources, and then also to have that properly documented, right?
Sure.
So the hypothesis. The proposed the hypothesis will have a section for the hypothesis. There'll be a short section for how you envision it being tested in the laboratory. Again, it doesn't have to be a full, sort of grant proposal write-up of, materials and methods. Needs to be more of a sort of conceptual framework as to how this would be tested in the lab. And then the documentation of the data resources you used, what processing you did. Again, that's to sort of help the judges, determine, again, how well the resources were used, what resources were used, and how they were used. And so.
Okay, great. Thank you. I think that was helpful. I think my main question there was about the testable, if you need to know the exact cell line, that this needs to be tested on, and these kinds of conditions. But it seems that this is kind of more just conceptual, as you mentioned.
Right. And again, the cell line, potentially may not exist. It might be something that has to be derived. So again, it really depends on the hypothesis, that gets developed.
Okay, thank you.
I'd like to just ask, I'm just going to ask a couple people to sort of give a little background. We'll start with Dogus Dogru.
Hi. Can you hear me?
Yes.
Yes.
Hi, I am first year grad student at Boston College.
Okay.
Under the supervision of Dr. Altundish. So what should I mention more? I don't know. We are working on viral proteins, mimicry of human hormones, and how they can be effect on diabetes patients.
Okay.
I'm not sure if you can see me or not.
Yes.
Oh, you can see me?
Yes.
Oh, okay, perfect. So I'm happy to be here.
Okay, great. And how about Carolina Setula?
Hello.
Hi.
Can you hear me?
Yes.
Well, we are from Argentina,
Okay.
and thank you for letting us participate in this challenge. We are from Buenos Aires, and I work at the Immunoendocrinology, Diabetes and Metabolism Lab at Austral University. So we are looking forward to participate in this challenge and to learn.
Great. Thank you, Carolina. Magdalena, do you want to introduce yourself?
Yes. Hi, everybody. My name is Magdalena Potok. My background is in epigenetics, and also I've done some bioinformatics. Currently, I work in a pharmaceutical company in oncology. I am very interested in participating, and I'm so happy that I'm able to, because my 5-year-old son has type 1 diabetes. He was diagnosed about a year and a half ago. And so I want to learn more about this disease and kind of use what I have learned in my training to use some of the bioinformatics approaches to find something that may help.
Fantastic. Thank you very much. How about Rob Rubino? Oops. You're Rob. I'll unmute you. Ask to unmute you. Rob?
Sorry, I'm eating lunch right now.
Oh, no worries.
Hi, my name is Rob Rubino. I'm a researcher affiliated with Dr. Leonardo Ferreira at MUSC. I'm excited to partake in this opportunity because I just recently graduated college, undergrad, so I'm fresh out of the boat, so to speak, but I'm excited to apply some of the skills I learned in my undergrad because I took an art class and used bioinformatics. And I've had previous research background in cancer metabolism and immunometabolism. So I'm excited to learn more about immune tolerance and autoimmunity and with type 1 diabetes.
Fantastic. Yeah, no, this is a very interesting group, very interdisciplinary group, and very diverse, which is fantastic. So
yeah, we have a high school student.
Okay, great. Erica, Heather Thumb.
Okay. Can you hear me? I don't know if you can
Yes.
see me. Can you see me?
No.
Oh, okay. Let's see. I might. Well, let me just go ahead. I don't want to hold things up. So I signed up to participate in the novice track. I have a background in mechanical engineering, actually, but I just started a program through the University of Florida in microbiology and cell science to learn more about to learn more about the science. I have a daughter who was diagnosed about 3 years ago with type 1 diabetes, and it just really changes, your life and your child's life. And so I just wanted to take this opportunity to learn as much as I can. I really am excited to participate.
Fantastic, Erica. Welcome. How about Miranda? Miranda Orellano?
Hello, can you hear me?
Yes, I do hear you.
Hello. Thank you. I'm on the same team with Carolina. We are from Argentina.
Okay.
And we are in Buenos Aires, Argentina, and both of us are PhD students, and that's a great opportunity for us to be in this challenge. So thank you so much.
Fantastic. And last but not least for today, although as I think everyone knows, we've extended our registration through October 1st, so we're encouraging more people to join. Luz Andriane, where's Luz, so I can. Oh, I don't see Luz here anymore. Maybe she's part of the other team. Okay, well, I think that's, it's great. It's great to meet you all. Again, spread the word. We are extending the registration through October 1, and there's plenty of room for especially novice track individuals. So, please don't hesitate to join us, and we hope you do. Jeff, is there anything else you'd like to say or contribute?
No, I mean, we had the email address, the dchallenge@dkNET.org. So, I mean, Monica will be setting up sort of the Slack space. I don't know, if people have used Slack, it's a good way to communicate, with the organizers. Then again, there's the email. So, if you do have any questions, don't hesitate to contact us.
Sounds great. We look forward to working with you in the months ahead. Thanks everyone for participating, and we'll see you soon. Bye for now. Hope you have a great rest of your day.
Thank you.
Thank you.
Thank you. Bye-bye. Okay. I think people are heading out. I'm gonna end our presentation, and I'll talk to you, Jeff and Koei, later.